The creeping silence of a conversation turning into a muffled hum is a deeply personal and often isolating experience. One may find themselves nodding along, pretending to understand, the vibrant world of sound slowly fading behind a frustrating veil. But what if the source of this slow fade wasn't just noise or age? What if the key to reclaiming clarity was hidden not in the ear itself, but within a network of forgotten microscopic highways that nourish it, and a single nutrient that keeps them open? This gradual decline in auditory sharpness is far more than a simple inconvenience. It is a slow withdrawal from connection. It's the missed punchline of a grandchild's joke, the fading melody of a favorite song, the growing distance in a conversation with a loved one. Each misunderstood word can feel like a small severing of a social tie, leading to a profound sense of isolation that can quietly permeate every aspect of one's life. For decades, the conventional narrative has been straightforward. Hearing loss is the inevitable consequence of a lifetime of loud noises and the simple wearing down of delicate machinery. This is, of course, a significant part of the story. The delicate hair cells, known as stereocilia, which are responsible for translating sound vibrations into electrical signals for the brain, are indeed fragile. But this explanation is incomplete. It fails to answer a critical question that researchers have been pondering for years. Why do some individuals, even those exposed to similar environments, maintain sharp hearing well into their 80s and 90s, while others experience a significant decline much earlier? The answer, it turns out, lies deeper than the surface-level damage. It resides in the silent, invisible crisis happening within the body's most intricate plumbing. This changes everything. The inner ear is not a passive piece of hardware. It is a living, breathing, and incredibly hungry ecosystem. The cochlea, that tiny, snail-shaped organ, is one of the most metabolically active parts of the entire human body, demanding a constant and uninterrupted supply of oxygen and vital nutrients to function. Deprive it of this fuel, even for a short time, and its performance begins to suffer, its cells beginning to weaken and wither. So, if it's not just about the external wear and tear, what is truly choking off this vital lifeline to our sense of hearing? And more importantly, is there anything that can be done to reopen it? The human body contains over 60,000 miles of blood vessels, an astonishing circulatory network. Yet the vast majority of this network consists of microscopic vessels, capillaries, and arterioles so narrow that red blood cells must pass through in single file. It is within these microvessels that the real work of delivery and exchange happens. Nowhere is this microvascular system more delicate or more critical than in the inner ear. This fragile network is the sole provider for the auditory cells. It is their only source of food, their only source of oxygen, and their only way to dispose of metabolic waste. When this system is robust and flexible, sound is clear and vibrant. The cells are nourished, healthy, and responsive. However, over time, this delicate network faces a relentless assault. A process known as oxidative stress, driven by factors from environmental toxins to normal metabolic function, generates unstable molecules called free radicals. These free radicals act like microscopic wrecking balls, damaging the fragile endothelial lining of the microvessels. This damage, in turn, can trigger a low-grade, chronic inflammatory response. The result is a gradual stiffening and narrowing of these vital passageways. They lose their youthful flexibility. The flow of blood becomes sluggish and restricted. This is a condition known as microvascular ischemia. It's a slow, silent starvation. The auditory cells, deprived of their lifeblood, can no longer perform their duties effectively. They begin to malfunction and, eventually, they begin to die. This is the true, hidden culprit behind so much of what is considered age-related hearing loss. It is not a sudden event, but a slow, creeping drought that unfolds over decades. The sounds don't just vanish one day. They slowly lose their richness, their texture, their clarity, as the cells responsible for perceiving them become progressively undernourished. This is a profound realization. It shifts the focus from simply protecting the ear from the outside to actively nourishing and supporting it from the inside. The question then becomes not, how can one avoid noise, but how can one restore the health and integrity of these microscopic lifelines? The answer was hidden not in a high-tech laboratory, but in the nutritional wisdom of cultures known for their remarkable health span and longevity. For centuries, certain traditional diets have been rich in a specific class of powerful plant-based compounds. These compounds were not seen as medicine, but as an integral part of daily nourishment. Modern science is only now beginning to understand the profound effects these substances have on the human body, particularly on the vascular system. These are not vitamins or minerals in the traditional sense. They are bioactive compounds, powerful antioxidants, and anti-inflammatories that work at a cellular level. Research emerging from institutions around the world has begun to demonstrate their remarkable ability to support the endothelium, the delicate inner lining of all blood vessels, from the massive aorta to the tiniest capillaries of the inner ear. They work by quenching the fire of oxidative stress and calming the chronic inflammation that causes vessels to become stiff and constricted. In doing so, they help restore the natural flexibility and permeability of the microvessels, allowing for improved blood flow and a more robust delivery of oxygen and nutrients. It is, in essence, a way of revitalizing the body's own internal delivery service. This class of compounds, broadly known as polyphenols, represents a paradigm shift in how one might approach preserving hearing. Instead of a passive defense, it offers a proactive strategy to maintain the very infrastructure that keeps the auditory system alive and functioning at its peak. It is about feeding the foundation of hearing. But within this vast family of thousands of different polyphenols, scientific investigation has revealed that a few are exceptionally potent in their effects on microcirculation. But among even these powerful compounds, one stands entirely alone, a forgotten key to unlocking these microscopic pathways. The problem? Modern food processing and agricultural practices have systematically stripped this specific, crucial nutrient from our diets almost entirely. It's a nutrient our ancestors consumed daily, yet one that most people today will go their entire lives without ever getting in a meaningful amount. This compound, which ancient texts referred to with names that translate to river of life or stone's blood, is now known to science by a far less poetic name, C-89 or cynarin-8 glucoside. But what is it exactly? And why has it been so completely erased from our world? Is its absence simply a tragic accident of agricultural efficiency? A casualty of our quest for bigger, sweeter, more resilient crops? Or is the truth something far more unsettling? The story of its disappearance and its recent rediscovery begins not in a boardroom or a farm, but in the dusty, forgotten archives of a university library, with the work of one man, a brilliant and by all accounts difficult biochemist named Alistair Finch. In the late 1970s, long before the mainstream buzz around antioxidants and polyphenols, Finch was obsessed with a single, radical idea: that certain plants didn't just contain protective compounds, but that they held specific molecular keys designed to unlock dormant regenerative pathways within human cells. He believed it wasn't about passively defending the body, but about actively reminding it how to heal itself. His colleagues dismissed him as a romantic, a mystic masquerading as a scientist. While they were busy isolating individual vitamins, Finch was studying ancient agricultural texts and obscure botanical folklore. He was convinced that the secret to vascular health, the key to keeping the body's tiniest rivers flowing freely into old age, was not found in a dozen different superfoods, but was concentrated in one specific family of plants and in one specific molecule within them. He found it in the wild, bitter thistles and cardoons that grew on Mediterranean hillsides, the ancestors of our modern artichoke. This C-89 compound was abundant in these tough, ancient plants. Finch published a series of small, dense papers. In them, he presented evidence that C-89 didn't just neutralize free radicals, it did something far more profound. It appeared to trigger the cellular production of a protein called NRF2, which is often called the CUC, master regulator, of the body's antioxidant response. Think of it this way. Most antioxidants are like fire extinguishers. They put out individual fires, the free radicals, one at a time. It's a crucial but reactive process. But Finch proposed that C-89 wasn't the fire extinguisher. It was the fire chief, waking up the entire cellular fire department, activating hundreds of the body's own protective and detoxifying genes. This single action created a cascade of positive effects. The chronic inflammation in the microvessels would subside. The endothelial lining would begin to repair itself. The vessels would regain their youthful elasticity. Finch theorized that a consistent dietary intake of this one compound could, over time, essentially reverse the process of microvascular stiffening. He called it vascular retuning. His work was ignored. It was too complex, too far outside the one vitamin, one disease model of the era. And then, his research came to an abrupt, silent end. He lost his funding, his lab was reallocated, and he faded into academic obscurity. The river of discovery had run dry. But why did his work, and the nutrient itself, vanish so completely? Here lies the truly shocking part of the story. It wasn't a conspiracy, it was a coincidence of progress. A quiet catastrophe that no one ever noticed. As agriculture modernized in the 20th century, the goal was simple. Yield. How can we grow more food, faster, on less land? Plant breeders began to select for traits like size, sweetness, and pest resistance. The wild, bitter thistle drive Finch studied was selectively bred over generations into the large, palatable, and thornless globe artichoke we know today. But in breeding out the bitterness, the very thing that signaled the presence of these powerful defensive compounds, they inadvertently bred out the C89 molecule almost entirely. The same story was repeated across the board. The ancient, nutrient-dense varieties of our food were replaced by their blander, high-yield, high-sugar descendants. We engineered the medicine right out of our own food, and we never even knew it was there to begin with. We traded cellular protection for a longer shelf life. We traded vascular resilience for a sweeter taste. So for 50 years, Finch's papers gathered dust. The memory of C89 faded. And all the while, the slow, silent epidemic of microvascular ischemia, the creeping drought in our inner ears, continued unabated, now accepted as a simple, unavoidable consequence of getting older. But the story doesn't end there. In 2019, a team of researchers at a European Food Science Institute were using advanced spectrometric analysis to create a molecular fingerprint of heirloom plant varieties. They were simply trying to catalog what had been lost. When they analyzed a rare, preserved sample of the ancient wild cardoon, the same plant Finch had studied, their machines lit up. They had detected a polyphenol with a molecular structure and weight they had never seen before in such concentrations. Intrigued, they dug into the literature. And there, buried in an obscure, pre-digital database, they found it. Alastair Finch's research from 1978. It was all there. The NRF2 pathway, the theory of vascular retuning, the profound implications for age-related conditions. Finch had been half a century ahead of his time. Armed with technology he could have only dreamed of, the new team was able to replicate and validate his findings in a matter of months. They confirmed that C89 was, without question, the most potent natural NRF2 activator ever documented. Its effect on microvascular endothelial cells was staggering. In lab models, it didn't just protect the cells, it stimulated them to regenerate. It was like discovering a blueprint for rebuilding the entire irrigation system from the inside out. The rediscovery of C89 and Finch's forgotten work sent a quiet shockwave through the research community. It was a revelation. Age-related hearing loss wasn't necessarily a permanent sentence of decay. It was a symptom of a specific, identifiable nutritional deficiency. A deficiency that we had, ourselves, engineered into our own food supply. The implications were enormous. But it immediately created a new, and perhaps even more difficult, problem. The science was there. The proof was in hand. But the source was gone. The ancient thistles that produced this compound in meaningful amounts were now exceedingly rare, found only in a few protected biospheres. They couldn't be mass cultivated without losing the very stressors that made them produce the C89 in the first place. You couldn't just plant 1 billion of them and solve the problem. The solution was right there, tantalizingly close, yet locked away by the very march of progress that had hidden it for so long. They had found the lost key. But could they now find a way to produce it at scale? To get it from these forgotten wild plants into the bodies of the millions who so desperately needed it? The greatest challenge, it turned out, was not one of discovery, but of delivery. For 3 years, the consortium of scientists hit a wall. Every attempt to synthesize C89 in the lab resulted in an unstable, inert molecule. Every effort to cultivate the wild cardoon in controlled environments failed. The plants grew, but the precious compound simply wouldn't form without the precise, harsh stressors of its native habitat. It seemed the miracle was doomed to remain a footnote in a scientific journal. But what if they were asking the wrong question? What if the goal was never to harvest it from a plant at all? In a final, desperate attempt to find a clue, a research assistant was sent to the university archives to retrieve Finch's original, physical lab notes, not just the published papers. And buried at the bottom of that box, beneath decades of forgotten data, was something else entirely. Not a lab notebook, but a small, leather-bound personal journal. And the entries inside painted a picture not of failure, but of a secret, terrifying success. Finch hadn't just discovered what the molecule did. He had discovered how to make it. Not in a plant, but inside the human body itself. Why was this revolutionary breakthrough hidden? Why did Finch's last entries speak not of triumph, but of a terrible new door he was afraid to open? Finch's journal revealed the true secret. The cardoon thistle didn't create C89 on its own. It was a symphony. The compound was the end product of a symbiotic relationship between the plant and a specific, non-pathogenic bacterium that lived in its roots, a microbe that thrived only in the harsh, nutrient-poor soil of its native environment. The plant provided the raw materials. The microbe performed the final, magical act of synthesis. This was why lab synthesis failed and cultivation failed. They were only looking at half of the equation, and this was the source of Finch's fear. He had isolated the microbe. He theorized it could be introduced into the human gut microbiome, where it could, in theory, produce C89 internally, turning the body into its own medicine factory. But in 1978, intentionally colonizing a human with a novel bacterium was the stuff of science fiction. He was a botanist, not a god. The Gazette, terrible new door, wasn't a failure. It was a success so profound, he felt it was too dangerous for humanity to handle. He buried his work, choosing caution over a discovery that could change the world. But where Finch saw peril, the new generation of scientists, armed with a deep understanding of the microbiome, saw a breathtakingly elegant solution. They could create a safe, stable, targeted probiotic. No rare thistles to harvest. No complex molecules to synthesize. The answer wasn't in a factory, but in life itself. They could reintroduce a missing partner to our internal ecosystem, a partner lost when we sterilized our soil and sanitized our lives. They had solved the problem of delivery. The story of C89 isn't just about hearing loss. It's a story about listening to the quiet wisdom of the past, to the forgotten corners of nature, and to the intricate biological conversations happening inside our own bodies. We engineered a deficiency, and for a time, we accepted the silence that followed as inevitable. But we've learned that the systems of the body don't just want to break down, they want to rebuild. We just have to give them the tools they lost. The blueprint for our own restoration is often found in the very wildness we sought to tame. The key wasn't a new drug, but a forgotten relationship, a partnership between plant, microbe, and man. It's a powerful reminder that the most advanced technology we will ever know is the natural world, and our own profound ability to heal from within. The journey back to health is not about finding a magic bullet, but about restoring a lost balance. The greatest discoveries are often not about finding something new, but about understanding something that has been there all along. The journey to uncover the truth and unlock human potential is never over. To continue exploring the science that is changing our lives, subscribe to Better_Health.